Hello, everybody. On the behalf of the Biochemical Society and Portland Press, I am pleased to welcome you to this webinar, which is part of the Biochemistry Focus webinar series. My name is Camilla Schele, and I will be your chair today. I am Associate Professor at the Novo Nordisk Foundation Center for Basic Metabolic Research at the University of Copenhagen. And in my research group, we focus on human brown fat crosstalk. So I am extremely excited about today's webinar on novel frontiers of adipose tissue in health and disease. This webinar will address current research and emerging concepts of adipose tissue biology in health and disease, and parallels a collection on this topic in the Biochemistry Journal, which is guest edited by today's speakers, Dr. Emilio Mutillo and Professor Gregory Steinberg. You can view this collection on the Biochemical Journal website, as shown on the slide. Some of the concepts that will be discussed in today's webinar includes the heterogeneity and the different types of adipose tissue and how they each are implicated in metabolic disease and therapeutic purposes. Now, before we start the presentations, I would like to encourage you to please type your questions in the box as shown in the image on the screen, and please state who your question is for. After the talks, I will ask your questions to the speakers. Our first invited speaker today is Professor Gregory Steinberg. Dr. Steinberg is a professor of medicine at McMaster University, where he holds a Canada Research Chair and a J. Bruce Duncan Endowed Chair in Metabolic Diseases. He is also the founding co-director of the McMaster Center for Metabolism, Obesity, and Diabetes Research. His research studies cellular energy sensing mechanisms and how endocrine factors, lipid metabolism, and insulin sensitivity are linked and contribute to the development of obesity, type 2 diabetes, NASH, cardiovascular disease, and cancer. He has published over 180 papers, many of which have been in the world's leading scientific journals. These discoveries have also spurred the development of numerous new classes of medications that have been approved or are in clinical testing for NASH and CVD. He has also supervised over 30 graduate students and postdoctoral fellows, many of whom are now clinicians, have faculty positions, or are research scientists within industry. The impact of his scientific contributions have been recognized by the Royal Society of Canada, the Endocrine Society, the American Diabetes Association, Diabetes Canada, and the Canadian Institutes of Health Research, who have each presented him with outstanding or early career scientific achievement awards. So with that very impressive track record, it's my pleasure to hand over to you, Greg. Take it away.
Okay, well, thank you very much, Camilla, for that very kind introduction. Can you hear me okay?
Yes.
Yes, okay.
You're all set to go.
I'm all set to go. Okay, good. Well, thank you so much for everyone for joining this webinar. You know, it was a real pleasure guest editing this special issue for the Biochemical Journal on Adipose Tissue with Emilio. And I want to thank all the authors who contributed to the special issue for writing such excellent reviews, which really made our job quite easy. Today, I wanted to provide a little background about sort of a big picture overview of how adipose tissue fits into disease development, and then highlight 4 review articles which fit into that theme. And then I'll hand it over to Emilio, who will really get into the nitty-gritty of the molecular circuitry which controls those pathways. And so, why do we care about adipose tissue, and what's the connection with disease development? Well, the most obvious connection is really the worldwide overweight and obesity epidemic. which affects over 1 billion people and has been growing rapidly around the world over the last 2 decades. And of course, why do we care about obesity? Well, obesity is tightly associated with many chronic diseases, but most notably type 2 diabetes. And there's now over 500 million people who have type 2 diabetes around the world. And as I mentioned previously, it's just not type 2 diabetes that is linked with excess adiposity or obesity. We can link up a whole host of new other chronic diseases which have an impact or which are impacted by obesity and increases in adipose tissue. Some of our favorites that we're interested in studying are fatty liver disease, type 2 diabetes, and cardiovascular disease, but you can really think of a whole host of diseases. And also, there's many different types of cancer which are linked with the development of obesity. So clearly, it's a major health problem. So this really gets you thinking, why is obesity linked to the development of these chronic diseases, and specifically type 2 diabetes? And so if we think about the normal glucose control, which is very tightly regulated by insulin typically, which stimulates glucose uptake into muscle and fat while suppressing glucose production from the liver, with the development of type 2 diabetes and insulin resistance, the ability of insulin to do this job is impaired. So what is the key triggers mediating that? Well, obviously it's not a simple answer, but there's an overwhelming a lot of evidence suggesting that there's 3 main factors. And one of them is the ectopic lipid storage within tissues which normally don't have a lot of fat in them, such as muscle and liver, which then in turn can trigger the development of inflammation and defects in mitochondrial oxidation, potentially linked to reactive oxygen species or other incomplete oxidative products, have all been linked to the development of insulin resistance or the inability of insulin to increase glucose uptake into muscle and fat and suppress hepatic glucose production. But one of the key really perplexing things, though, and potentially why adipose tissue was overlooked for so many years, is because if we look at this equation, fat in humans is a relatively very modest player in the regulation of glucose control with respect to the uptake of glucose from the circulation, impacting around 10 to 20% of total glucose disposal at a maximal level. So it really doesn't come across as why would we focus on adipose tissue if we were trying to link it to the development of type 2 diabetes. However, this was a fairly simple view at the time. And we now know that as adipose tissue expands with obesity, there's a whole host of things that happen and that can communicate with the rest of the body. And in this sort of outdated and simplified schematic, we discussed some of these issues at this time, where with the overnutrition, adipose tissue expands, macrophages and other immune cells infiltrate the adipose tissue. This indeed in turn leads to dysregulated adipokine secretion, and indeed hormones such as adiponectin, leptin, resistant RBP4 are some of the examples whose activity are altered. And these hormones can directly impact the hypothalamic centers critical for regulating energy expenditure, food intake, and hepatic glucose production, which obviously leads to a feedback loop leading to increased overnutrition and continuing expansion of the adipose tissue mass. In addition, we can have defects in free fatty acid or potentially alterations in the ability of insulin to suppress free fatty acid-induced lipolysis, and of course inflammation. And all of these things can lead to the development of liver insulin resistance, inflammation, ectopic lipid accumulation, elevations in plasma glucose, as well as insulin resistance within the muscle, in turn reducing the development of glucose intake of glucose into the muscle, the primary site of glucose disposal. So in this way, adipose tissue is really central to the regulation of glucose control. So in this issue of the Biochemical Journal special issue, we've tried to link this with other conditions, and it provided sort of an updated view on how adipose tissue may be central to the development of insulin resistance and type 2 diabetes. And the first article I wanted to highlight here today was by Dr. Scherzer and Dr. Barra, who talk about the NLRP inflammasome. And specifically, we're all well aware of this inflammatory pathway leading to the activation of NF-kappa B and the induction of those cytokines that I mentioned in the previous slides, such as IL-6 and TNF-alpha. However, in their review, they add increased scope to this classical cytokine, IL-6 and TNF, by also describing in great detail the interleukin-1 beta pathway and specific and how this activation occurs in response to many lipid moieties such as ceramides, mitochondrial defects, and other lipid species, which can in turn, following the priming step, which is induced by classical cytokines such as IL-6 and TNF, can lead to the cleavage and activation of the IL-1 beta pathway. Importantly, they discuss in further detail how this pathway can contribute to the development of insulin resistance by suppressing insulin signal transduction, can lead to adipose tissue fibrosis, a big factor leading to the health of the adipose tissue. We know if we impair fibrosis and allow adipose tissue to continue to grow healthy, we don't develop insulin resistance, and also angiogenesis within the fat. So this really highlights, the interconnections between metabolism, immunity, and disease development, and how NLRP3 may be critical for that process. In addition, it leads another key concept here is that disease can directly regulate immunity, and immunity can regulate metabolism. And this is based off of studies where statins, which are used to treat cardiovascular disease, can lead to the activation of the NLRP3 activation in IL-1 beta production, which in turn leads to the development or promote the development of type 2 diabetes, which we know is associated with statin intake. So in this way, disease and metabolism may be directly linked through the NLRP3 inflammasome. Another key question tackled through this special issue is, what are the causes potentially for the increased incidence of obesity? And we're all aware of this relationship where we have this classical scale where there's a dysregulation between energy expenditure and energy intake, most notably reductions of physical activity and increase in caloric intake of high calorie foods is associated with the obesity epidemic. But perhaps it's not quite that simple. And there's growing body of evidence to suggest that environmental chemicals within that we are exposed to through the air, through the water, and through the food may have an impact on our metabolism. In particular, there's growing evidence appreciating the role of agricultural production processes and potentially how these influence adipose tissue. In particular, pesticide use we know has increased dramatically over the last several decades, and all these pesticides may potentially accumulate in adipose tissue given their lipophilic nature of many of them. And so in this review, Dr. Holloway, in collaboration with Robert Getskill and Evelyn Sakaridis and myself, discuss some of these pathways by which different classes of pesticides may impact adipose tissue biology. And these were focused on potentially mitochondrial pathways, key pathways regulating adipogenesis, insulin sensitivity, adipokine production, and inflammatory cytokine production, all of which have been shown to be influenced by these different classes of pesticides. Of course, a key question and challenge, though, we found when reviewing the literature is that, modeling environmental exposure to primary to adipocytes in animal models was really limiting in many of these studies. And most of these studies were done at high concentrations, potentially using the 3T3L1 model, which is obviously not an ideal model given its immortalized nature and fibroblast background. And there's also many in vivo studies which really made it difficult to delineate effects of these pesticides on obesity versus direct effects on adipose tissue. So really not fully understanding whether these things were having direct effects on the adipose tissue. Lastly, there is really a paucity of information about how these pesticides might affect brown or beige adipose tissue thermogenesis. So lastly, one of the ways we know that exercise, we can tackle insulin sensitivity and improve insulin sensitivity in type 2 diabetes is through exercise. And this data is well known from the diabetes prevention program. And commonly we think about these studies improving muscle glucose disposal and insulin sensitivity in the muscle as the primary factor driving these effects. However, there's emerging evidence to suggest that adipose tissue may in fact be very important for some of these metabolic adaptations to exercise training. And to understand this fully, we need to think about the different types of fat within the body. We have classical brown fat within the adipose tissue, the intrascapular depot within the rodent, which is expressed as abundantly uncoupling protein 1. And then we have a beige fat group, which is really a hybrid of both the white and brown fat. And, the beige fat is special because it can recruit uncoupling protein in response to cold or other adrenergic stimuli to really take on this hybrid between the white and the brown fat. And of course, as one of the key defining features of both brown and beige fat is that they both express uncoupling protein 1, and as such, you can generate a futile cycle in which the electron transport chain is decoupled from oxidative phosphorylation, thus inducing a large number of induction of the TCA cycle and glycolytic pathways to try to maintain ATP ratios within the cell. And then as a result, heat is generated. Of course, one of the key factors driving this pathway is the brain, and we know that the brain senses cold, and that this signal transduction pathway results in the release of norepinephrine, which at least in the rodent binds to the beta-3 adrenergic receptor, which can in turn stimulate lipolytic cascades through activation of pKa, induction of fatty acids, and hydrolysis of fatty acids, which activate uncoupling protein 1. In addition, we've shown in collaboration with Emilio, who was the first author, and Camila, who did studies in the human brown adipose tissue, that adrenergic stimuli also activate AMP kinase, and this is really important for promoting the browning of white fat and mitochondrial biogenesis in the cell. So ultimately, this generates heat and may protect from the development of fatty liver disease and insulin resistance. In their article, Dr. Wright and Greg McKee discuss some of the mechanisms and biochemical adaptations which occur in white adipose tissue following aerobic exercise. It's been recognized for many years that the exercise induces lipolytic cascades, but in this article, they go into further detail about how these lipolytic cascades may be very important for the browning of white fat, at least in rodents, and how these signaling pathways potentially induced by catecholamines, interleukin 6 or other glucocorticoids or corticosteroids may induce lipolysis, and this in turn generates free fatty acyl-CoAs, which are then reesterified. And this reesterification process is an energy-intensive process requiring ATP, which may induce an energy charge deficit in the cell, which in turn activates AMP kinase, stimulating transcriptional coactivation and coactivation of PGC1 alpha, which in turn induces the mitochondrial browning program within the white adipose tissue. Of course, we also know now recently that fatty acyl CoAs can directly activate AMPK, so this may also be important for some of these effects. However, that remains to be directly tested. However, it does suggest that, at least in rodents, that some of these effects may be important for some of the insulin sensitizing effects that we see with exercise. And they review some of the literature around that. And in some instances, the lack of translatability of these findings to human studies where browning has not been observed in some depots with exercise training and what that means. And of course, that's really what we're interested with all this work. A lot of the work has been done in rodents and cell lines. And the key question really is, can any of this be relevant to humans? And if so, how do we measure it? And this takes us to the last article that I'll discuss today by Dr. Blandin and Dr. Richard from the University of Sherbrooke, where they detail functional characterization of the brown adipose tissue depots. And in particular, they discuss how these depots can be regulated. And here showing in this diagram, we can see that the brown fat is activated in response to cold in humans, and this can be picked up using different tracers, in this case FDG, and that these depots can be activated much like rodent depots. So indeed, humans do have brown fat. They also discuss different methodologies which can be used to measure brown fat activity using different radio tracers within humans to assess, depending on what you're interested in, different aspects of brown adipose tissue metabolism. And they provide detailed explanations for how these different tracers may be utilized, for example, to measure innervation processes, hemodynamic changes, mitochondrial content, metabolic rate, for instance, using the acetator, water, oxygen probes, or whether the triglyceride lipolysis may be measured using MRS or MRI technologies. Lastly, they provide a comprehensive overview of, comparing relative changes in brown adipose tissue activity between mice as well as adult males and females, and the potential contribution this activation of brown adipose tissue may have on oxygen consumption rates, VO2 max, percent of VO2 max in response to mild cold, and changes in substrate utilization rates, which really leads us to potentially question whether activation of brown adipose tissue thermogenesis in humans may be a viable therapeutic target for treating metabolic diseases. So in summary, these four articles I've discussed within the Biochemical Journal special issue on adipose tissue have really focused on how adipose tissue, the inflammasome, regulates adipose tissue metabolism, how the impact of pesticide exposure on adipose tissue development and function could influence adipose tissue development, and in turn could potentially lead to cardiometabolic disease. How biochemical adaptations in response to exercise could influence adipose tissue metabolism and may be potentially important for improving insulin sensitivity. And lastly, in the final article discusses the potential implications of these findings to humans and how we can potentially monitor these changes using various methodological and imaging approaches which have been developed to visualize or measure brown adipose tissue metabolism. So with that, I'll hand it over to Emilio to discuss some of the more detailed studies regarding the molecular cues which are critical for regulating brown adipose tissue development.
So thank you very much for this excellent talk, Greg. And to all listeners, please remember that both speakers will answer your questions after the presentations. So our next invited speaker is Dr. Emilio Motillo, an assistant scientist in the Hypertension and Cardiovascular Research Division at Henry Ford Hospital in Detroit, Michigan. The overall goal of his research career has been to understand the molecular and cellular mechanisms that regulate metabolic homeostasis, with a long-term goal of translating basic research findings into therapies for human health. His work over the past 12 years has led to discoveries that excessive lipids can cause inflammation, that lipolytic products can act as signaling molecules, and the discovery of mechanisms by which brown adipose tissue is dysregulated during obesity. He was awarded an NIH Pathway to Independence Award, where he has expanded on his skill set to utilize lipidomic, proteomic, and imaging approaches to determine the lipid mediators, phosphorylation events, and signaling pathways that are dependent on lipolysis. So with that introduction, I would like to hand over to you, Emilio. Please take it away.
Great, Camilla. Thank you for that kind introduction. So for the rest of the talk, we'll be focusing on the more molecular and cellular aspects of adipose tissue biology, as Greg mentioned, and this brings really some fundamental questions that are being addressed in the rest of our issue in this biochemical collection on adipose tissue biology. And one of the questions that arises from what Greg talked about in terms of this conversion of white fat to brown fat is, how is this plasticity regulated? How can we better understand this conversion of browning of WAT, and by further understanding that, how can we target it therapeutically? So it's one of the fundamental questions that will be addressed in our review issue. Secondly, how are lipids released from adipocytes during this process, biochemical process of lipolysis? How do fat cells release fatty acids at the molecular level, and potentially how do these lipids signal to mediate various metabolic effects? Another fundamental question that arises is, what is the molecular mechanism for heat production? We know that UCP1 is an important mechanism, but are there other mechanisms that exist that might be potential sources of heat production and can be potentially targeted therapeutically? Finally, an outstanding question is, how do we therapeutically target adipose tissue? And with our final review article, we'll see potentially how that might be done. So in terms of understanding the plasticity of adipose tissue, at least from rodent models, we know that there's this conversion of white fat to brown fat that can occur under chronic adrenergic stimulation. So if we subject mice to chronic cold exposure, we can convert white fat to this brown fat-like phenotype, as seen here in these various depots. And to better really understand how this occurs, various techniques have arisen, such as genetic reporter techniques that rely on identifying a specific genetic marker and then tracing those cells in vivo to see if those progenitor cells can convert to a brown adipocyte, or potentially under conditions such as high-fat diet, if they can convert to a white adipocyte phenotype. And in addition to other more traditional techniques, such as perspective analyses, such as FACS, where we can isolate these progenitor cells and then see if they differentiate in vitro into a brown adipocyte or white adipocyte, or potentially by transplanting them back in vivo to see if they can produce a fat cell. So the problem with these techniques is that they're limited to a specific genetic marker and really don't give us a comprehensive view of what's happening at the cellular level and understanding how these potential stromal non-adipocyte cells interact with the fat cells to mediate tissue homeostasis. And so in the review by Rondini and Grandeman, they highlight the use of single-cell RNA-seq to deconvolve cellular heterogeneity in adipose tissue. So in this technique of single-cell RNA-seq, barcoded beads are mixed with single-cell suspensions, such as cells isolated from adipose tissue, where they form a single oil droplet containing a barcode, where we can barcode our single cells and subject these single cells to sorting and analysis at the single cell level, such as reverse transcription, fragmentation, PCR amplification, providing really a global overview at the gene expression level, still at the single cell level. This is followed by sequencing, demultiplexing, and alignment, where we can arise with this cell classification at the single cell level, which allows us to identify potential novel cell populations that reside within adipose tissue, or potentially identification of novel adipocyte progenitor cells, such as ACS1 cells or ACS1A cells, identify proliferating, differentiating cells, depending upon the expression of proliferating markers. And again, allow us to identify novel cell types that might reside within adipose tissue, such as cells that have a mesothelial expression profile. And again, this can be very dynamic, in that this process can then be subjected to analyzing tissue under various conditions, such as cold exposure or high-fat diet, or understanding how these single cell dynamics might change under conditions such as diabetes. And secondly, in the review by Pollard and Carlin, that highlight how these single cell techniques, in addition to the techniques such as genetic tracing and prospective FACS analysis, work together to give us a view of the stem cell niches that might exist within adipose tissue, where these techniques have allowed us to understand that within the fascia and mesothelium of adipose tissue, there resides these early progenitor cells that seem to give rise to these adipocyte progenitor cells, such as ACS1B, ACS1A cells, that can then potentially commit to a white adipocyte given the correct cues. And again, we have other cells that compromise adipose tissue, such as immune cells, which seem to play an important role in eliciting these cues to push adipocytes to a committed phenotype. In addition, these techniques have allowed us to identify cells that reside within the vascular endothelium that express various markers, such as the PGF receptor markers, which can give rise to brown adipocytes depending upon adrenergic stimulation, and that can potentially even commit to a beige adipocyte. And not only has our understanding improved at the cellular level, but also at more the genetic and molecular level, understanding the important transcription factors that control the thermogenic program. So in the review by Haesuk Sol's lab and Dr. Yi, they really go over the epigenetic dynamics that occur during the thermogenic program that are important in committing cells to the brown adipocyte or thermogenic program. This involves this big assembly of proteins that are involved in promoting the expression of genes such as USP1. So we have important DNA binding transcription factors that are recruited to these DNA elements, such as C-terminal binding protein, CBP beta, PPAR gamma, an important target for the anti-diabetic medication TZDs. Other novelly identified DNA binding elements, such as zinc finger protein 516 and other elements that can bind upstream of the promoter regions. In addition, these transcription factors can recruit known co-regulators, such as PGC1 alpha, a well-known master regulator for mitochondrial biogenesis. Then also PRDM16, a master regulator for commitment to the brown adipocyte lineage. And these cofactors that can then recruit DNA modifying enzymes, such as the epigenetic modifiers, LSD1, which is a demethylase that can demethylate the DNA to open up these chromatin where they can be subjected to gene transcription, and additional regulation by the histone deacetylases that can deacetylate PGC1 to promote its activation. And finally, many of these factors can be phosphorylated. So this pathway is really in queue with various external factors that might occur in terms of cell surface receptors that might activate these this gene transcriptional program. And so a better understanding of this gene transcription might allow us to find novel targets in committing cells to this thermogenic program and potentially recruiting more brown fat that can be used therapeutically. And so another fundamental question that arises is, how is adipocyte metabolism regulated? So some of the more seminal works from the 50s and 60s really identified adipose tissue as the main site for lipid storage, where studies by Hausberger showed that with insulin treatment, adipose tissue was this main site for the storage of lipid as measured through lipogenesis. And then work in the 60s went on to show that adipose tissue was the main site for the mobilization of fatty acids through this process of lipolysis, where these fatty acids can be mobilized and used as an energy source. So here Rod Bell showed that treating isolated fat cells with various hormones led to the release of free fatty acids and a net change in triglyceride levels. And this view of adipocyte lipolysis has really greatly increased over the past 40 years, where now we have begun to understand it as a very dynamic process that is regulated by various protein interactions that occur on the surface of lipid droplets. And in this review highlighted by my own group with Alexander Yang, we highlight these dynamic protein interactions that occur. So in a basal state, you have a protein such as ABHT1, which is a lipase coactivator protein, which is normally found on the surface of lipid droplets bound to this scaffold protein called perilipid 1. And then we have the rate-limited enzyme for triacylglycerol hydrolysis, PNPLA2, which under basal conditions has very weak activity. However, upon stimulation of receptors such as the beta-adrenic receptors, which is important in stimulating thermogenesis in brown fat cells, this leads to phosphorylation of various proteins, in which this allows a release of ABHT5 from perilipid 1 to bind to and greatly activate greatly stimulate the activity of PNPLA2, where now we can fully activate this lipolysis process, where now these fatty acids can be oxidized within mitochondria and act as a ligand for UCP1 to promote thermogenesis. And not only are these fatty acids substrates, but work from our group and that of others has gone on to show that these fatty acids can actually act as signaling molecules, where we've been able to show that fatty acids can traffic to the nucleus and bind various transcription factors such as the PPAR nuclear receptors to really mediate a transcriptional program involved in adipocyte differentiation, or further upregulating a transcriptional program involved in fatty acid oxidation and thermogenesis, thereby further increasing uncoupling protein content and further generating more heat. And not only does lipolysis signaling occur at this really molecular level, but work from many groups has uncovered that lipolysis can also signal at the whole body level under various and regulate various aspects of physiology. So adipocytes are known to secrete bioactive lipids in response to lipolysis, such as the FAFAs. These FAFAs can bind various receptors found on the liver to suppress gluconeogenesis. Fatty acids released by fat cells can activate PPAR alpha in the liver to promote VLDL secretion. Fatty acids generated by lipolysis can also stimulate afferent nerves, where in the brain this increases sympathetic output to further increase brown fat in thermogenesis. Brown fat is known to secrete bioactive lipids that can further promote fatty acid uptake in thermogenesis. And indeed, fatty acids can stimulate receptors found on islet cells to promote insulin secretion and insulin action, thereby resulting in this whole body response to fatty acid release. And finally, these adipocytes can also secrete extracellular vesicles that can carry various protein and RNA factors to signal to other cells to mediate metabolic responses. And however, lipolysis can also become dysregulated under various pathological conditions such as obesity and diabetes, where the excess release of fatty acids from fat cells can lead to the buildup of these lipid intermediate products such as diacylglyceramides and ceramides, which Greg highlighted upon, promoting this lipotoxicity in the liver, which has negative effects on insulin signaling and thereby promoting insulin resistance in the liver and thereby increasing gluconeogenesis also in the liver. In addition, these fatty acids released by adipocytes can instill an insult on macrophages, whereby this results in further increase of inflammatory cytokines that can have negative effects on adipocyte metabolism to further promote fatty acid release, really initiating this cycle of back and forth release of fatty acid and cytokines between the fat cells and macrophages that has a negative impact on adipocyte metabolism and whole body tissue homeostasis. Furthermore, cancer cells can release various factors that can act to stimulate lipolysis, and whereby this promotes lipolysis from fat cells, and these cancer cells can now use these fatty acids as fuel to increase growth and metastasis. Indeed, these cancer cells are also known to secrete various extra cellular vesicles that can also promote lipolysis to also fuel this metastatic and cancerous growth. So really, targeting the adipocyte lipolysis is really a double-edged sword, where under pathological conditions, it seems that the best way may be to inhibit lipolysis. However, under physiological conditions, if we could find ways to further stimulate these homeostatic mechanisms, such as PPAR alpha or these GPCR receptors, we can potentially find a way to regain lipid homeostasis under these various pathological conditions. So another question that comes up is what is the mechanism for heat production? So as Greg highlighted, UCP1 is this well-known mechanism for heat production found in brown fat. And so UCP1 creates essentially this futile cycle by acting as a leak in the inner mitochondrial membrane, whereby it's activated by fatty acids, thereby creating this futile cycle that consumes ATP and thereby generating heat. However, the much interest in understanding UCP1 and its function led to people making the UCP1 knockout mouse, and it was somewhat surprising upon generation of the UCP1 deficient mouse that this mouse was normal for adiposity, so had normal body mass, where the initial thought was that these mice would be obese due to the fact that UCP1 was this important mechanism for burning energy and generating heat. However, these mice showed normal body weight. And further studies went on to show that these UCP1 knockout mice can be slowly acclimated to the cold, where they can actually survive in the cold, and led to the idea that there was this existence of these UCP1 independent mechanisms that functioned to maintain body temperature of these mice and allow them to survive in the cold. And so in the review by Rosler and Kazak, they highlight the UCP1 dependent mechanism and many of these novel UCP1 independent mechanisms that have been uncovered, such as the creatine phosphocreatine cycle that cycles within mitochondria to use up ATP and in turn generating heat in this process. Additionally, we also have the constant cycling of fatty acids and the reesterification that occurs in adipose tissue, which is also an energy demanding process. And through this energy demanding process, this also subsequently generates heat. In the ER lumen, we have the circa ryanodine calcium cycle, where the constant cycling of calcium between these two pumps results in the consumption of ATP and also heat generation. And finally, the authors touch upon the ATP-ADP carrier system, whereby it may create a leak in a mitochondrial membrane that is sensitive to fatty acid stimulation to potentially also act as a heat-producing mechanism. So potentially through the discovery of these novel UC-independent mechanisms, it may provide additional methods that we can target therapeutically to promote energy wasting processing, potentially as anti-obesity treatments. And finally, this brings us to our last question, and that is, how do we therapeutically target adipose tissue? So in rodents, we have a pretty good idea of doing this, and that's through the activation of these beta-3 adrenergic receptors, which again are found on the surface of brown adipose tissue. So these receptors under physiological conditions are activated by norepinephrine, which is released in response to cold. And we have a good panel of drugs developed in such as the beta-3 adrenergic agonist, which can selectively target the receptors in the rodents. And activation of these receptors are known to have potent anti-obesity and anti-diabetic properties. However, whether this is the case in humans has been unclear for some time, as the therapeutically targeting the beta-3 adrenergic receptor in humans has been problematic. And so in recent findings from our own webinar chair, Dr. Camilla Chile and Denny Blondin, they go on to show that in human brown fat, they do not express the beta-3 adrenergic receptor, but instead, thermogenesis seems to be mostly mediated by the beta-2 adrenergic receptor. So in this case, the beta-3 adrenergic receptors have no effect on thermogenesis in brown fat. However, norepinephrine stimulated thermogenesis in human brown fat, and by knocking out the beta-2 adrenergic receptor, this effect was shown to be abolished. However, this brings us to another problem: is that how do we do this? How do we activate brown fat safely in humans with the fact that beta-2 adrenergic receptors are found throughout the body and the heart and other organs? This may pose potential problems. And so in the final review by Dr. Usar and Onugi, they review the various methods to identify cell surface receptors found on adipocytes and progenitor cells, and potentially how we can characterize these receptors and target them therapeutically. And so they summarize various indirect techniques such as RNA analysis and transcriptomics to identify cell surface receptors found on fat cells. Other techniques such as co-immunoprecipitation to enrich cell surface receptors found on fat cells, followed by proteomic analysis. And then more direct approaches such as the use of cyclic peptides or nanobodies and aptamers to also find cell surface receptors that might be expressed on adipocytes, where these receptors can then be further characterized and tested in tissue or in mice to see what metabolic effect activation of these receptors through these various approaches might have. And so in this review, they highlight the various receptors that are known to mediate thermogenic programs in adipocytes, such as FGF21, receptors that are known to stimulate glucose uptake in adipocytes, such as the insulin receptors and FGFRs, receptors that are known to stimulate lipolysis on fat cells, and also those that stimulate lipogenesis. And they also summarize the various surface epitopes that are found on the pre-adipocytes, and that those that are pro-adipogenic and also anti-adipogenic. So potentially understanding how we can target these receptors may provide potential other avenues to recruit novel fat cells, improving tissue homeostasis in adipose tissue, or potentially finding ways to other ways to increase the thermogenic program in brown adipose tissue. So this brings us finally to some further outstanding questions that remain in the field. And that is one of the questions that remains is really how relevant are these rodent studies these rodent studies on obesity and thermogenesis to humans. The fact that we reside at thermal neutral, many of the experiments that are performed in rodents are not performed at thermal neutral, but instead at 23 or 25 degrees, where instead thermal neutrality for a rodent is 30 degrees. So should we really be performing these rodent experiments at 30 degrees, considering that we actually every day reside at thermal neutrality? Finally, is browning of wet a valid therapeutic option in humans? What is this plasticity in humans? We know that we can get this interconversion between white fat and brown fat in rodents, but is this a plasticity occurring in humans? And I think hopefully we'll be able to further understand that in the future. Finally, this brings us to another question: that how do we safely target brown adipose tissue? And again, with the advent of novel imaging techniques that were highlighted by Denny Blondin's group, we may begin to understand how we can target and measure brown fat activity in humans. Finally, an outstanding question that remains is, what impact do environmental factors have on obesity? We know that genetics play an important factor in determining obesity and diabetes, but obviously there's interaction between various environmental factors that were highlighted by Greg, and understanding how these environmental factors interact with genetic factors to promote obesity and diabetes. And so in closing, I'd just like to highlight our adipose tissue biology collection, where you can find out further information on these reviews and all the contribution of the authors discussed in our webinar today. I thank everyone for attending, and I think we will take it over to Camilla.
Thank you very much, Emilio, for this excellent talk. So now we can welcome questions. We already have a lot of questions coming in, but you can still do that while we are starting this question session. So let me start. So the first question actually came in already during Greg's talk earlier. So this is about the disconnection between type 2 diabetes and obesity. So what might be the reason for increased type 2 diabetes rates in India, even when obesity rate is low in Indian subcontinent? And I direct this to you, Greg.
Yeah, that's a great question. You know, I think it's well known that Southeast Asians have a greater propensity for type 2 diabetes at a lower BMI, and you know, this can actually be directly potentially attributed to adipose tissue storage, where Caucasians are more equipped with potentially storing fat, you know, in subcutaneous depots compared to visceral depots, and as a result, the risk of developing type 2 diabetes is lower or higher in India, given potentially at a BMI that we don't classify as obese. And I think, you know, that's the simplest answer that I can think of for that. Obviously, the genetics leading to those changes are very interesting and need to be explored further.
I mean, in the regards of your observations on pesticides, do you think that could also be a contributing factor in this case?
Absolutely. You know, I think you know, it needs to be explored, certainly, whether there's some interaction there, whether there is differences to pesticide exposures or other environmental exposures, that maybe some chemicals have been banned in Europe and other parts of North America where they're still heavily used in India. It could be one possibility for sure. So that's a great point, Camilla.
Okay, so now one for you, Emilio. Can fasting control the growth and heat generation of adipocytes?
So fasting, that's a good question. Yeah, so fasting probably mediates a different response in that it mobilizes more external lipid stores rather than activating brown fat. So I think there's literature suggests that during fasting, brown fat is not activated. So it's probably activating different receptors and potentially pathways that might be blocking brown fat. While there are beneficial effects to fasting in terms of you know, intermittent fasting approaches that are thought to be beneficial. it's thought that these probably don't really relate to brown fat per se.
Yeah, great. Yeah, great point. So, moving on to you again, Greg. Can liposuction decrease the chance of the onset of type 2 diabetes if the person is not affected with it till now?
Liposuction, yeah. So I'm pretty sure that's been studied. My guess is that we do know from examples, say, of lipodystrophy, if you don't have a sufficient fat stores, you are very insulin resistant because you develop fatty liver very quickly. And so, potentially a pronounced amount of liposuction, you may increase your risk of developing type 2 diabetes. I'm sure it's been studied.
Yeah, so it's the opposite basically. So it's the opposite from, I mean, the question was actually if it can decrease the chance of the onset, but probably
it should increase.
Yeah, because then you lose your safe storage of the lipids, basically. All right, so
bad idea.
Move on. It's not a good. I've been thinking about this. Why someone should stop this? It's not a good idea. I mean, it could be dangerous if you can't. I mean, it would be better to take away some other organs, I think. Yeah. All right. So, is there any evidence linking the exposure of chemicals such as BPA or food additives such as monoacylglycerols on browning and brown fat thermogenesis?
Yeah, we didn't, there isn't much evidence linking BPA used with brown fat or food dyes with brown fat. There's very few chemicals which have been tested in brown fat or brownable cell lines. In fact, almost all the pesticide and environmental exposure studies, as I mentioned, have all been done in the fibroblast 3T3L1s, which have no browning potential. So that's a very important question to reapproach a lot of these toxicological studies with cell lines which are brownable or have more brown like progenitors. So it's really unknown, I would say.
Okay. So over
one
to
one of the complications is that all these studies are done in vivo, and if they're affecting appetite and other things, it can also really change things without affecting the adipocyte directly.
Yeah, sure. So over to you, Emilio. A couple of questions from Kusum Karbanda. Is there a role of PLIN1 in HSL activation? And is there a direct role of FAP4 signaling, or is it just a protein for fatty acid transport?
No, that's a great question. So I think in terms of those molecular players that are involved, I think we have a pretty good understanding of those proteins and interactions. So PLIN1 is very important. If you have, for example, there's mutations in humans that have mutations in PLIN1, and these patients are lipodystrophic and have metabolic disease. So it's a very important protein, and so I think it's important in organizing those interactions. And so as I've shown that studies that lack PLIN1, you have aberrant lipolysis and potentially mistargeting of some of these proteins such as ABHT5 and HSL, which might not be beneficial. So I think there is strong evidence for understanding the role of PLIN1. And then with regard to the fatty acid binding proteins, I think there is some literature suggests that they do have an important role in signaling and potentially carrying these fatty acids not only to the nucleus but potentially to other compartments, so acting as these lipid binding proteins and shuttling these ligands and mediating these signaling events. Of course, that might depend upon the tissue types. With the fatty acid proteins that are expressed in various tissues, this might differ upon the tissue type.
Yeah, I mean, I think this is very interesting. The role of fatty acids as signaling molecules. I mean, is that something growing? I mean, it's something that we will know much more about in the near future, or what is your thoughts around that?
I think
I mean,
understanding.
you mentioned the signaling to the brain even. I mean, as
Right.
activator of the sympathetic
Right.
nervous system.
Right, right. So it's not only the fatty acids, a lot of lipids that mediate a lot of signaling events, and we're just beginning to uncover that with advances in lipidomics and imaging studies. I think understanding how lipid homeostasis is regulated, a key would be to understanding how these lipids might mediate these homeostatic responses. So I think there's a lot more to be learned in terms of understanding how lipids might act as ligands for other proteins, or potentially co-activators for other proteins, or allosteric activators for that matter. For example, AMPK being one example. These long chain CoAs can activate AMPK, and potentially that could be part of it too.
Yeah. Okay. So I see that there's a brown fat related question here, so I will move on with that. It's also for you, Emilio. How is the quantitative contribution of the various wasting cycling mechanisms to basal brown fat energy expenditure?
So I guess that depends if you're talking about humans or rodents. So we know that rodents have substantial brown fat. So probably USP1 would be the major mechanism there. But depending upon the cold stress and the length of the cold stress, obviously potentially these other mechanisms can be recruited, and obviously they become a bigger player in mice that are deficient for USP1. In humans, I think we really need to understand a lot more. Depends how much brown fat is there in a human to begin with, and potentially do these other mechanisms also exist in humans, and if they do, can we then target them? So I think there's a lot more to be done on human side of that, while the rodent models do provide a good model for understanding these USP1 independent mechanisms, and mice are not the same as humans. So I think by studying things more on the human side, we'll be able to get at those questions a bit more.
Yeah, completely agree on that. Right. So yeah, great answer. So now over to you, Greg. Some questions about leptin and adiponectin. So what is the trigger for increased circulating leptin but decreased adiponectin levels in obesity? And then there's a related question here. Do these adipokines have a paracrine role as well? And do adipocytes secrete pro-inflammatory cytokines? So that was a few questions in one.
Yeah.
Let's start with first.
That's a good idea. So, I think, the leptin levels are, really tightly linked to the size of the adipocyte. The bigger the adipocyte grows, the more leptin mRNA is expressed. The exact mechanisms linking that, I don't recall exactly, and the inverse is really seen with adiponectin, where not necessarily the size of the adipocyte is important, but it appears to be the sensitivity of the adipocyte to insulin. Any insulin sensitizer will improve adiponectin release, and obviously the most obvious example of that is with rosiglitazone, which expands the adipocyte and increases adiponectin secretion, but also at the same time improves the sensitivity of the adipocyte to the insulin. So, insulin seems to be a major insulin sensitivity, major driver for adiponectin cell size, major driver for leptin. And so the last question about the inflammatory cytokines, I mean, I think, the studies done around in 2003, really linking it to a lot of the inflammatory cytokines coming from macrophages. But, since those initial studies, we know that, adipocytes also have neutrophils, T cells, you name the immune cell type. They live in adipocytes, they come in there at different stages, and some of them come in and recruit the macrophages. So, most likely these other immune cells, these professional immune secreting cells are probably the primary source for inflammatory cytokines, I would say. is still the most well accepted model. And they in turn can affect adipocyte secretion of adipokines, classical adipokines, such as adiponectin.
Yeah, I mean, I think it's also, it's also some of these proteins that are, it depends a lot which context they are secreted in, if they are pro-inflammatory or not. I mean, for IL-6, for example, it's such a multifunctional protein, so it depends on the context around it and the tissue status around it. So yeah, it's a complex situation, I guess.
And we didn't get a chance to talk about your favorite adipokines, badokines.
That will be next time, maybe.
Next time,
webinar. Yeah. All right. So I'm getting reminders here that we should start to wrap up this great webinar. So I guess I have to do that. So I would like to then thank both of you speakers. It was really a true pleasure to hear and see your presentations. And I would also like to thank all the participants for being so active with questions. And I mean, we didn't have, unfortunately, not have time to answer all the questions, but the discussion can go on online. There is a Twitter address, two Twitter addresses that you can follow, and you can continue the discussion there. And as a final remark, I have also been asked by the Biochemical Society to mention some housekeeping statements. You can see here on the slides as well. So the Society welcomes suggestions for future topics and speakers to feature in this Biochemistry Focus webinar series. So you can find more information about the webinars and propose your webinar on their webpage, as you can see on the slide. And if you're interested, please join the next webinar in this series. The title is Neurobiology of Chronic Pain, Mechanisms, Management, and In Between. It's Thursday next week at 3 o'clock. And you can find out more about the Biochemical Society, a community of researchers and specialists, by visiting their website. And so thank you once again, and take care, everybody.